Introduction of Microanatomy Mini Project. Histological techniques play a crucial role in aquatic science by allowing researchers to study the microscopic structure of aquatic organisms and their tissues. In this experiment, we explored the fundamental procedures involved in histological sample preparation, including tissue collection, fixation, embedding, sectioning, staining, and mounting. By gaining hands-on experience with these techniques, we aim to understand how they contribute to our ability to investigate the anatomy and physiology of aquatic organisms. Through this exploration, we will develop essential skills for studying the intricate biology of aquatic ecosystems and their inhabitants.
At the end of the experiment, we were able to learn the basic technique of histological sample preparation, including tissue collection, fixation, embedding, sectioning, staining, and mounting. We were able to understand the effect of the different fixation and staining method on tissue preservation and visualization under a microscope. We then developed a proficiency in performing histological procedures and interpreting the resulting tissue sectioning for microscopic analysis. At the end of the experiment, we were able to learn the basic technique of histological sample preparation, including tissue collection, fixation, embedding, section.
Next, we move to the methodology part, which is in attaining histology of fish tissues. The method was started with sample collection and continued with sample fixation, dehydration, embedding, cutting, staining, and mounting. So the methodology part were initiated with the sample collection. So the sample that were used in this part here is a Bristol-Lepis fasciata, and the standard length, total length, weight were recorded. So the gill, skin, gonad, liver, heart, and intestine were dissected into a small pieces. And then we move to the sample fixation. So the organ sample that were dissected earlier will be placed into the separate leopard vials contain formaldehyde as our fixative solution. Next, the methodology process will proceed with the embedding and the cutting. So during the embedding, the paraffin wax will be heated and poured the melted wax into the mold, and the sample tissue will be pressed gently into the semi-solid paraffin block and submerged mold in the cold water to prevent paraffin crystallization. And all of the wax samples will be kept in the cold plate histoco for 1 week. So for the cutting process proceeds by the using microtome for sample trimming and sectioning. So in histology, a microtome primary function is to precisely cut tissue samples into extremely thin, uniform section for microscopic examination. Meanwhile, fishing method also were applied to split the ribbon onto the slide. The section were then dried for above 30 to 60 minutes to prevent detachment during the staining. For the second last methodology in attaining the histology of fish tissues is staining and mounting. So the process of staining in copolene jar with hematoxylin and eosin are as follows and were executed under the fume hood. So in histology, staining is a crucial technique that uses dyes to color tissue components, enhancing their visibility and contrast for microscopic examination. So the staining process divided to two parts, which is cleaning and rehydration and staining. And chemical use were xylol, alcohol with different dilution, which is 100%, 95%, 85%, and 70%. Tap water and eosin were used in this process. So each chemical has their own timing soak for each sample. And then we move to the mounting process, which is after staining. We need 2 to 3 drops of resin to be dropped onto the slide for mounting. The sample then were covered with the cover slip, and the slide will live in the warmer condition for at least 24 hours. And the sample slide were ready to be observed under the microscope for the histology microscopy session.
This is sample S1, showing the gill tissue under histological observation. At 40 times magnification, we can identify the gill filament which support the overall structure of the gills. Extending from these filaments are the primary lamellae, and from them the thinner secondary lamellae branch out, increasing the surface area for respiration. Under the 100 times magnification, the finer details become visible. The epithelial lining that cover the lamellae can be clearly seen, along with the nuclei of the epithelial cell stained dark purple. Running through the secondary lamellae are the capillaries, which facilitate gas exchange between the water and blood. This microscopic view demonstrates the efficient design of the gills for oxygen uptake in aquatic environment. This is sample S4, showing liver tissue observed under histological staining. Under 40 times magnification, we can see the general structure of the liver. The hepatocytes, or the liver cells, are arranged in cords radiating outward from the central vein region. Surrounding these hepatocytes are sinusoidal spaces, which appear as clear gaps and allows blood to flow through the tissue for the exchange of the substance. At 100 times magnification, we can get a clear view of the hepatocytes nuclei, which appear as a dark purple round structure within its cells. The cytoplasmic detail of the hepatocytes are more defined, showing a pinkish granule texture. We can also observe the lining of the cell of sinusoids, including endothelial cells and a possible Kupffer cells, which is a part of the liver immune function.
So for the sixth result, which is larvae, this is the histology image on 100 time magnification captured under the microscope. They were observed that there were outline of developing organs, muscle segmentation, and early vertebrae. And it is visible to us for see it here, which is the bone, the eyes, the mouth. Toward a final result, which is gonad of the fish specimen, there were two magnifications that were captured, which is 40 times and 100 times, similar with the previous one. So under the 40 magnification, we can see the seminiferous part of the testis, and zooming into the seminiferous under 100 times magnification, it can be seen that there were secondary spermatocytes and primary spermatocytes of the fish specimen.
Problem occurred during this experiment are sample damage during fixation. During the fixation process, the tissue sample was accidentally damaged. This may have been caused by improper handling, prolonged exposure to the fixation, or incorrect fixation conditions. As a result, the sample lost its original structure, making it difficult to proceed with proper sectioning and staining. This compromised the quality of the final slide and affected the accuracy of the observations. Second, overcutting during microtomy. While sectioning the tissue using the microtome, the sample was overcut, resulting in sections that were too thin or damaged. Overcutting can cause the tissue to tear, fold, or produce uneven slices, which make it challenging to mount the sample properly on a slide. This error often requires repeating the sectioning process, leading to time loss and possible depletion of the sample. Third, mislabeling or misarrangement of samples. There was an incident of incorrect labeling or misplacement of samples during the experiment. This led to confusion in identifying the tissue sections and disrupted the flow of the analysis. Proper labeling is essential in histology to ensure traceability and accurate comparison of results. So this issue significantly affected the reliability of the data collected. For the discussion, histological preparation and methodology. In this experiment, histological techniques including fixation, dehydration, embedding, sectioning, staining, and mounting were employed to study fish organs at the microscopic level. These techniques are essential to preserving tissue architecture and cellular integrity. Fixation with formalin is widely used for aquatic histology. If tissues are not fixed promptly or adequately, autolysis and degradation can occur, leading to structural loss. Hematocilin and eosin staining was used, which is effective for visualizing general tissue architecture. Hematocilin stains cell nuclear blue, while eosin stains cytoplasm and extracellular structures pink. While ideal for routine scanning, it lacks specificity for certain cell types, which can be resolved using specialized stains such as PAS or Asian blue. Next, for the interpretation of fish organs. The first one, gills. Histologically, fish gills displays primary and secondary lamella, with thin epithelial layers crucial for gas exchange. Gills are among the first organs affected by waterborne contaminants. Damaged lamella, hypertrophy, or epithelial lifting may be observed when handling or fixation is poor. Next, liver. The liver serves detoxification and metabolic functions. It normally presents as polygonal hepatocytes arranged around sinusoids. Poor fixation or excessive sectioning, as seen in our experiment, can distort liver architecture. Histopathological analysis of fish livers is commonly used in environmental monitoring. Next, gonad. Fish gonads are vary with reproductive cycle phase. Ovaries typically contain developing oocytes, while testes have layers of spermatocytes. Accurate sectioning and proper staining are vital to distinguish these stages. The presence of immature or atretic follicles can also suggest environmental or hormonal disruptions. The final one is larvae. Larvae tissues are extremely delicate and prone to damage during embedding and sectioning. Larvae histology often focus on organogenesis, including the development of swim bladder, intestine, and musculature. Successful visualization is critical for aquaculture development and early disease detection. For discussion of fish organs, gills show normal lamella. These are sensitive to pollutants and can be easily damaged. Poor fixation can affect how clear these structures appear. For liver, had typical structure, but sectioning issue caused some damage. The liver is important in detoxification and is often used to check for pollution effects. Next, gonads. Gonads show reproductive cell abnormalities. Here can signal hormonal disruption in polluted environments. And final one is larvae. Larvae were fragile and often damaged. Larvae tissue is important in aquaculture to study development and survival. For analysis, evaluation of histological process. First one is fixation errors. Tissue degradation due to delay or improper fixation was noted. This can lead to artifacts or misinterpretation under the microscope. A fixation ratio of at least 1 equal to 20 and correct fixation type are key factors in tissue preservation. Next, overcutting during microtomy. Overcut section result in ultra-thin, fragile tissue that tears or folds during mounting. The optimal section thickness is 4 to 6 micrometer for fish tissues. Calibration and skillful use of the microtome are necessary to avoid this issue. Final one is staining effectiveness. While H&E provides an excellent overview of tissue structure, specialized staining can better reveal details such as mucopolysaccharides or glycogen. This could improve analysis, especially for intestine or gonad tissue. For functional relevance of tissue, each organ's microstructure reflects its physiological role and environmental vulnerability. Gills are exposed to the aquatic environment and reflect real-time water quality. Livers show cumulative effects of toxins and diet. Gonads provide reproductive health indicators. Larval histology is key in development biology and early pathology detections. These insights are vital for fields like fish farming, environmental toxicology, and aquatic veterinary science.
This experiment successfully demonstrated the fundamental histological techniques required to analyze fish tissue at the microscopic level. Despite the challenges such as fixation error, overcutting during microtomy, and sample mislabeling, students gained practical insight into tissue preservation, staining, interpretation. The microstructure of gills, liver, gonads, and larvae revealed essential biological function and highlighted the importance of accurate histological procedures. These skills are valuable for application in aquaculture, environmental monitoring, and aquatic health assessments.